Okay, we back? Alright, people. So this is on, guys. It's lesson 2. For the people at home, it's lesson 2 under plant tissues. So we've got this double today. So what I've done, and I'm going to read it to you people because you're here, alright? They can read it. Okay, let's leave it like that. Okay, so what I've said here, in the one video that I'm going to show you now, the fancy word for any flowering plant is angiosperm. So roses, daisies, acacias, grass, orchid, geraniums at the back there, they're all angiosperms, even the long spiked plant, okay? And it's because angio means covered seed, sperma means seed. So the seeds are found, don't write anything down. The seeds end up being in a fruit, and then the fruit gets eaten or taken away, and it's covered until it can be dropped on the ground, and sand over it, and then it germinates again. Yes, not again, it germinates. Sorry?
How does that work with grass?
Okay, so if you mow your lawn and you get those little feathery things, those are the anthers and the, what do you call them? Anthers and the stigmas. The female part, the pistils, the gynecium, I don't know what you guys call them in grade 8. That's what's hanging out. So those feathery things lose the pollen and it lands in the stigmas. Those are the flowers. Okay, so yes, there's lots of different types of flowers, and flowering plants, angiosperms, make seeds, and they're protected in the fruit, and the fruits can be from tiny to really big, like a watermelon. Sorry, I've just got a tickle. Postnasals, nothing else. Okay, so then you also get gymnosperms. Let me just grab one here. Alright, so these things. Okay, so you get angiosperms and you get gymnosperms, and gymnosperms are like, they're conifers, they're pine trees. But this is a female cone, and the little seeds would be in here. In fact, I can see where the little seeds were, because they have little wings, all right? And I can see where they left their mark on top of these woody scales. And, oh, I must stand in front of there. All right, so in springtime, when the little yellow male cones come out and everyone gets hay fever? Those tiny little yellow male cones make pollen, and then it goes inside. These are very small at the beginning. They're probably this big, and then the pollen goes inside, and then these little scales close up, and then fertilization takes place, and the seeds are made, and then it takes 4 years for one of these female cones to grow. And this is the mature female cone, and the seeds will be in here, and they look like little I call them helicopter seeds. Right?
And then you go.
Yeah, but these are quite small, and they actually lie, each of these woody scales, it looks something like that, they lie like this, inside. Alright, so that's just for interest. Okay, we don't do it again. And they lie in here, and as these woody scales open up in the 4th year, then you get seed dispersal. So pine trees, they've also got seeds, but the seeds are protected in cones. So these little male cones that make all that yellow pollen, then I know spring is coming. And then these tiny little female cones, and they change color and shape. They close up when the pollen is inside, and then they change color and shape, and they get really big and woody, and then they produce the seeds. Okay, so that's just some general knowledge. All right. We used to do this in detail when I first started teaching government school, even private school. We still have a bit of it there, but we choose not to do it. We don't have to. We used to do this in detail: angiosperms, gymnosperms, algae, everything. It's not even part of the government. All of that is not even part of the government syllabus anymore. Okay, so from yesterday, we're trying to see, so how do plant tissues differ in a plant? And people go, oh, we're doing plants. It's not that bad. It's a story, and that's why, I'm going to show you that video that I made last night. That's why I keep going back to what is a plant cell. Think of a plant cell. Now, how's it going to be different in a root? How will it be different in a stem or a flower? It's easy. Flower, chromoplast. Do you remember? Color. Root, leucoplast, store starch. Leaf, chloroplast, make sugars. Okay. photosynthesis. All right, so that's what we're going to be doing. We're first going to look at the different tissues, and then we are going to try and put them together in we call it a stereogram, a three-dimensional diagram of a plant. And we're going to be colouring in and going mad. All right, so the people at home today, they will be watching me, well, you're going to watch it a bit now, talking about, this is a model of inside a leaf. I've got one at home. Luckily, we've got two, and I could take them home. And this is a model of the inside of a stem, but only one type of stem. Okay, so I'll show you now what I mean. Oh, I don't have enough space here. Okay, so what I've got here, just to show you that you know what's going on. So we're going to watch this video. Plant structure Bozeman. It's Mr. Anderson, but we're only going to watch the first 8 minutes, and there's lots of words there that you don't need to use, like monocotton, dicotton, angiosperm, etc. So we're just going to watch 8 or so minutes. Oh, I see it's all out of, I don't know why it does this. When I type it, it looks fine, and then I upload it, and then it's all like zigzag. Okay, so we don't need to worry about nodes and internodes, it's just interesting. Ram and Sam, he's talking about the root growing tip and the stem growing tip, the buds. Alright, and then you're going to watch the video that I made last night, the introductory one, because I don't want to repeat it, okay, because I think I might forget what I said there. And there's even more, but I'll explain in class using, and I don't think I'm going to make it. Alright, so let's go. Sorry guys, it wasn't like this.
And ma'am, I'm sorry, is this software an open book?
Yeah, it's open book for months. I didn't take ACC.
What is the software, sir?
It's on cells, cancer, mitosis. It's open book, it's just for fun. Just to make sure you've got it.
When is it though?
On Thursday, but I put things in.
Oh.
I put reminders in. I think we might have to turn off the light.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about plant structure. So a good way to think about this is plant anatomy. If you've never stood next to a giant sequoia, then you should. It dwarfs all the trees around it. It shows you how huge plants can become. Just using a few simple ingredients like water, carbon dioxide, and a few nutrients from the soil, you can become massive. And you can see the rings that we'll talk about kind of near the end. And so basically everything I'm going to talk about for the most part is going to be angiosperm, so flowering plants. And flowering plants can be broken down into two different types, dicots and monocots. And so what is a cot? A cot is simply a cotyledon. And so a cotyledon is going to be a baby plant leaf. And so here we've got two different seed types. There's going to be a seed coat around the outside, a little endosperm in here, but you can see this baby plant. And in this dicot, you'll have two cotyledons, 1, 2. But in a monocot, you're only going to have one. Now, the quintessential dicot that I think of is dandelion, and monocot I think about as grass.
Okay, so it's a little bit different to what I might use. So a dicot seed, think of a bean. Remember the one we used for mitosis? There's two parts, and the little embryo is there, and that becomes the leaf, and this becomes the root, and all of this here, the cotyledon, that's the food. That's what we eat for food. That's what that little embryo is going to use for food. So dicotyledon, there's two parts. Just for interest, monocot, I would probably draw a milliseed that we make popcorn with. So there's a little embryo, if it weren't all dried out, there's a little embryo at the bottom. You can't break a milliseed into two, so it's got one food storage area or cotyledon, and the little root will grow out, and the little leaf will grow out, and then it becomes a new plant. So angiosperms, you get monocots and dicots. But that's not the only difference. Even their leaves. If you look at grass, go sit on the grass today and look at the little veins going down the leaf. Parallel. Lots of, you can't see here, lots of little parallel veins. Net veins in dicots. Even the flowers. There's 1, 2, 3, 4, 5 petals, 5 stamens, 5 sepals. Remember from grade 8? All right, all combinations of 4, 4, 8, 12. But in monocots it's 3. So here there's, well, can't really see there, but 3 petals, 3 sepals, but they're the same colour as the petals, 3 anthers or 6 anthers, 1 stigma, but 3 parts at the top or something. Okay, so it's just what we used to do, and we don't do that anymore. Even the roots. Who pulls out weeds? And they've got those, no one. And they've got those long roots. So that's a dicot, weeds, plants that grow in your garden that you don't want them. Weeds. No one pull out weeds. Go and pull out some weeds. It's very relaxing. Okay? So there's the long roots, and it's got all these branch roots. Remember the pericycle? Okay, that makes branch roots. That's what dicots have. And then monocots. Pull out a piece of grass sometime. You see all those fibrous roots. They're all the same. Lots of little roots. Not just one main taproot with branch roots. He's going to say it now, but grass roots have, there's lots of fibrous roots. So those are all the difference between monocots and dicots. And we don't really do it anymore, but I just mention it now and then. So that you realize there's a difference out there.
And so if you've ever looked at a dandelion leaf, all the veins, which really are vascular materials, kind of like our circulatory system, moving water and sugar, are going to be net-like. They're going to branch out. But if we're talking about a monocot, they're going to be parallel. So if you've ever looked at the veins in a grass, a blade of grass, it's going to be all parallel. If you look at their flowers in a dicot, they're going to have 4 to 5, or multiples of 4 to 5 on their petals. So you can see 1, 2, 3, 4, 5. It's probably a dicot. Whereas if it's a monocot, they're going to be in multiples of 3. So you can see that this one has 6 petals, and so it's going to be a monocot. And then another way to differentiate between the two is going to be roots. A dandelion, if you've ever tried to pull one out, they have this really big taproot system. But in monocots, like grass, they're going to have a net-like root system. It's going to be what makes up sod, for example, in grass. And so those are the different types of angiosperms.
What's sod? Americans, when they fix up their house and their garden, you know the programs on TV, I watch them for just relaxation. And then they lay down the rectangular pieces of grass.
Yes.
A new lawn.
Yeah.
That's sod. We don't call it sod.
It looks like fake, or like
No,
you
it's
can buy it
real.
on the side of the road.
Yes, that's sod, yeah. Rolled up and you lay down the sod, and then you've got a new lawn. Okay.
When we talk about phytotomy, you should realize that plants live a double life. They have a life underground. We call that the root system, and then they have a life above ground. We call that the shoot system. Within that shoot system, basically you're going to have nodes. So that would be a node there, a node there. You have another node there, and another node up here.
Okay. So we used to do this as well. Nodes are the little regions where leaves develop, and there's all different patterns. Sometimes they're all around a stem. Sometimes, I need to stand here. Sometimes it's like zigzag, like this one. There's a node, there's a node, there's a node, there's a node. Sometimes circular, lots of leaves around the stem at one point. Or opposite. Have you ever seen branches where the leaves are opposite? Or like that, zigzag. Or it's where the little leaves, where that little bud is to make new leaves. The little, what do you call them? Oh, I've got a blank. Side buds, lateral buds. Remember that?
Yes.
Mitosis. Okay, so there's just so much variation. Go and sit on the grass and go and look at it sometime.
Here. But the distance between those nodes is going to be the internode. So that would be the internode between the two. And so basically plants are able to grow up, but they're also able to grow out at each of these node points. Just like us, they're going to have tissues. And so in us, if we're talking about tissues, now you would think of muscular, nervous, connective, and then epithelial. But in plants, there are just going to be 3 types of tissues. They have dermal tissue, ground tissue, and then vascular tissue. And just like us, they break that into specific types of cells. We've got the epidermis, which is the dermal lining. And then we have periderm, which is going to be mostly when we get to the level of bark, so it's secondary growth. Function of that is to provide protection. So this is a cross-section of a leaf, so it's going to provide protection from the outside. Same thing right here, you can see this in a stem. We're going to have dermal tissues on the outside. And they also prevent water loss. So basically epidermis is the big type of dermal tissue. Ground tissue is going to be just run-of-the-mill cells. And so this is going to be broken into 3 types, and these words are really fun to say: parenchyma, collenchyma, and sclerenchyma. What do? Basically, they do the jobs of the plant. So they're going to be the site of photosynthesis, for example, in the leaf, but it's going to be metabolism, storage, growth, all of that's going to be in the ground tissue. And then finally, we have the vascular tissue. That's made up of two types, xylem and phloem, and they're going to move the water and the sugar. So we'll get more specific to each of these. So let's start with the dermal tissue. Dermal tissue, for the most part, is going to be epidermis. So right here we're looking at a cross-section of a leaf. So this would be dermal tissue on the top and on the bottom. The guard cells also make up part of that. And the guard cells, you can see a zoomed-in version of it right here. Basically what they do is they surround the stomata, or this opening. What does the stomata do? You can see it's the hole in the leaf. Basically, it allows water to evaporate out, and that water, as it evaporates out, is going to carry water all the way up in the plant. But they also bring in a really important gas. That's going to be CO2. And so plants kind of have guard cells that are doing really good feedback. Basically, if they have a lot of moisture and they can let a lot of it go and it's really sunny, they open the guard cells up, and the stomata are going to allow a lot of water to come out, a bunch of carbon dioxide in, so they can make a bunch of sugars. Likewise, if it's really, really hot, really, really dry, they can close up the stomata so they don't lose all of their water. One thing that I should mention is going to be this real waxy covering on the epidermis. That's called the cuticle. And it's like wax. It's that wax that you feel on that slippery stuff you feel on the outside of a leaf. And that's going to prevent water from getting in and getting out.
Transpiration.
If you get to the ground tissue, basic run-of-the-mill cells are going to be parenchyma cells. The typical plant cells, parenchyma cells, what do? They're going to be the site of metabolism, site of photosynthesis. Outside that, as a plant starts to grow, we have the parenchyma. I always remember the C and the L, and parenchyma stands for celery. And so those are going to be these real durable cells on the outside of celery. They provide support as it starts to grow. And so you can see the dermis on the outside, parenchyma cells, and then these are going to be parenchyma cells right here. They provide support. And if you take a plant as it grows and just mess with it all the time, push it all the time, simulating like wind, the parenchyma gets stronger and stronger and stronger.
Okay, so I normally set this up. These are actually the veins. That's the xylem and the phloem, and the parenchyma, it's the strengthening tissue. It's just over here. It's really pretty to see under the microscope. And then you've got the epidermis surrounding the celery stem, and this in here, the parenchyma. Parenchyma means packing tissue. It's filling up the plant. That's what you eat. You're eating cellulose and, you're eating molecules. All right, so celery is quite nice to use. Ground tissue. What is the plant filled up with? Packing tissue, the juicy part, and then parenchyma and sclerenchyma, the stuff that makes it, celery grows tall.
But it never gets as strong as the sclerenchyma. Sclerenchyma are going to be the really durable, wooded kind of portions of the plant as it starts to grow. This is a sclerenchyma fiber that's cut in half. If we were to look at a fiber, we could find this. This is hemp. We pulled the sclerenchyma cells out to use these fibers to make rope. It's incredibly durable. And so sclerenchyma is going to be these big fibers that give it that really, really strong, like in a branch, that growth. But we'll also have sclerenchyma cells like the core of an apple, for example, is sclerenchyma. It's protecting the seeds on the inside, or the grit on the inside of a pear is sclerenchyma. So that's the ground. Now we go to the vascular. Vascular is going to be made up of two types, the xylem and the phloem. So in this, we're cutting a stem apart. This would all be xylem in here, and then level 4 right here is going to be phloem. Basically, xylem is moving water. It's going to move water from the roots to the shoots. Phloem is going to move sugar up and down in a plant. And so on the outside, we'd have dermis, and then we're going to have some sclerenchyma cells that are giving it durable support. So now we're going to talk about growth and then finally finish up with flowers. But this is an old story. If you go and take a spike and hammer it into a tree, let's say we put it at about 3 feet right here, and you come back in 100 years, so let's say, make it more realistic, 40 years, and the tree now, instead of being 16 feet tall, is 160 feet tall. How high is that spike going to be? Well, the right answer is it'll still be 3 feet tall, because plants grow from the top and they grow from the bottom. They grow from the shoots and the roots, but the middle is going to stay the same. Now the bark is going to start to grow up, and so that tree the spike might not be as far in. But just like humans, we first grow up, get much, much longer, and then I could tell you this, as you get older, you start to get wider and wider and wider. And so we call this primary growth, this first growth. And how does that occur? Well, we use something called an apical meristem. Basically, that's going to be a site. Right here, we're looking at a root where you have cells that keep copying themselves. We call those undifferentiated. Think of it like a stem cell that keeps making copies of itself. And so that's going to make new cells. And as those cells get longer, as they mature and get larger and larger and larger, that's going to push this stem or root in this direction. Now I can tell this right here is going to be a ram or a root apical meristem because it has this root cap on the top of it. And that root cap is going to allow it to push through the soil to find water. And so this meristem is actually going to produce new cells on this side, which will make the root cap, and then cells on this side that make this root itself. If you look at the SAM or the shoot apical meristem, it's not going to have this root cap because it doesn't have to, it's just pushing its way through air, so it's not going to have this, but it's still going to have this meristem because it's producing new cells behind it, and as they mature, it's bigger and bigger and bigger. So that's going to allow us to grow up and down, but we also have secondary growth.
Okay, so that's where I'm stopping. So he's just talking about the parts of the plant that we covered when we did mitosis. He's talking about the little meristems at the ends of the roots to make the roots grow longer and to make the little root cap to protect it like a little helmet. And he's talking about the meristematic tissue in the bud that gives rise to leaves or branches, or the meristematic, the dividing tissue that makes the plant taller. And then remember we said if we cut it here, then it starts branching sideways. Remember pruning?
Yes.
Okay, so there's a lot of stuff here. We'll get into that, okay, and we'll use nice diagrams and things to try and make sense of it. Would you like to watch the next one? Should we just carry on? Okay, so instead of me now trying to remember everything that I said yesterday, I'm going to show you. So, okay, let's look here. So there are the notes. I must give you the notes. So the notes, it's a table, then it's a photo worksheet. I'm going to give you that now. We've done that. This is my intro one last night, all right? Then I started explaining the table one, which I can do in class today. And then, so I did the table and I did a PowerPoint of the table. And then I put my actual PowerPoint in. So this video here is an introduction. Then me videoing, explaining the table on meristematic tissue. And me videoing, explaining meristematic tissue in my PowerPoint. And then this is my PowerPoint. Right, and then I've also got on Google Classroom, I'll need to just put all the notes in one section for this section, also it gets a little busy. Alright, so let's have a look here. Oh, wonderful. It's because it hasn't been done as a link yet. You know what that means? So I uploaded, I did it on my computer. So I've now uploaded it to Mr. Rosemez's file. Hang on, is there sound here? And then he gives me a link before it goes into the school's YouTube channel. I don't think it's going to work. So I'll just do it myself. So people at home, you can watch that if you want, or you can listen to me and then watch that at home. And you guys can watch it at home later. Alright, I thought that would work. It's not. Okay, so, alright, so what I did in that video, I took this, guys, I just need to get onto Google Meet, because one of the grade 9s is writing a test now. Let me just do that quickly. Won't you hand out the notes, and we need to punch them as well.
I'll have you that.
Yeah, so they are, while I'm trying to get onto Google Meet.
This is one of my parties. Yeah.
Hang on, where are they? Oh yeah, I'm in front of you. Alright, so there is a punch. Just do some of them, and I'll do this later.
How many of them? One?
Okay, let me just get onto Google Meet. Sorry people, I just need to get into grade 9.
One of each of these, huh?
Yes,
Just to 8. Okay.
yes. Yeah, you can go to the ladies. Okay, I'm coming now. Sorry, Noreen is talking to me.
Yeah.
if it's going okay. Alright, okay. And don't worry about, you've got an hour, but I mean it's a little bit after 10 now. So you'll finish at whatever, 11:05, and then you've got time to submit. Okay. Alright.
Thank you, ma'am.
Here we go. Okay, people at home, we are just handing out the notes. And we're going to carry on soon. I was hoping to show the video I made at home, but it's not playing because I need the link. Mr. Ruzmeza has to give me a link.